feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hello and welcome to the National University of Singapore Yong Lu Lin School of Medicine's Healthy Longevity webinar. Sorry, just making sure my volume was working. Thanks for joining us again. I'm coming to you from Berlin for the Rejuvenation Startup Summit is happening the next 2 days. So I'll be talking at that. So I'm in one of my favorite cities, and we'll have Colin Selman from Glasgow. So it's an all-European event tonight. Talking in a minute. I'd like to remind you to use the Q&A function to send questions. And before we get to Colin, we have Taejian Hua, a PhD student from the Center for Healthy Longevity, who will present on this project, the association of physical function and physical performance with epigenetic clocks in oldest old Singaporeans, the SG90 cohort.
Thank you for the kind introduction. As mentioned, my name is Jianhua, a PhD student in the NUS Center for Healthy Longevity. Today, I will be presenting my research on the association between physical function and performance and epigenetic age in the oldest old population of Singapore. To understand my research, first we have to define physical function, physical performance, and epigenetic age. Physical function refers to an individual ability to perform daily activities such as walking and climbing stairs. And physical performance refers to an individual ability to perform specific tasks such as hand grip strength tests. As we age, physiological changes lead to decline in strength, balance, and mobility. Why is this concerning? Research has shown that poor physical function in older adults is linked to 1.3 to 2.7 times higher mortality risk compared to healthier individuals. Next, epigenetic age is measure of biological age reflecting overall health status determined by analyzing DNA methylation patterns in the blood. DNA methylation involves adding methyl group to specific sites on our DNA, and there are 28 million methylation sites across our genome. Using elastic net regressions, researchers select a subset of methylation sites that strongly correlate with either chronological age or hazard of mortality. These are some of the epigenetic clocks used in our study. The main differences between them are the training dataset and outcome. The first generation was trained using chronological age data. The second generation was trained using hazard of mortality. Lastly, the third generation was trained using the pace of aging. If you notice, each of them has different numbers of methylation sites due to the differences in their training data regarding race, age range, number of subjects, and tissue types. Epigenetic clocks are promising health biomarkers. Studies have linked younger epigenetic age to physical activity, healthy diet, while accelerated epigenetic aging is associated with higher mortality risk, cardiovascular disease, and cancer risk. However, these findings are not well established in the oldest old Asian population, which refers to individuals aged 85 years old and above. Our study aims to investigate the association between epigenetic age and physical function and performance in this demographic. Linear regression revealed that green age outperformed other green age estimators. The result is presented in a heat map with blue indicating that a lower epigenetic age is associated with higher physical function, whereas red indicates the opposite. The asterisk sign indicates statistical significant association. The graph shows that lower green age is associated with higher physical function and performance in 5 out of the 9 assessments. Green age superior performance may be due to the fact that green age was trained to predict hazard of mortality. Furthermore, the training dataset for green age encompasses individual age up to 78 years old, in contrast to Denalian PACE, which was limited to a cohort of 45 years old individuals. Therefore, green age exhibits greater sensitivity in capturing muscular health compared to other age estimators. In conclusion, green age shows promise as an indicator of muscle health and functional decline. However, green age is currently only available as a research tool and requires further validation before clinical use. Randomized controlled trial and Mendelian randomization are therefore needed to establish causal links between green age and muscle measures. With further research, early detection of accelerated biological aging through green age could enable the implementation of personalized healthcare to mitigate physical decline, therefore contributing to the promotion of healthy aging and maintenance of independence in the oldest old population. I would like to acknowledge everyone for their contribution and making this research possible. Thank you for taking the time to listen. Should you have any further questions or require additional information, please do not hesitate to contact the Center for Healthy Longevity. Thank you.
Thank you. I think that this is another one of the studies that's really starting to validate these biologic clocks, and looking in the oldest old population is very important as we move forward. So a very interesting study. I want to introduce Colin Selman, who's a professor of biogerontology at the University of Glasgow. His research aims to understand the fundamental mechanisms underlying the aging process using a range of dietary, genetic, and pharmacological interventions, primarily in mice. So something near and dear to my heart. The topic of his talk today will be genetic pathways to a longer, healthier life. It's great to see you, Colin. It's been a while.
Yes, it has, Brian, a long, long time. I remember coming over to your lab when I was a MIA postdoc many moons ago in Seattle to do some polysome work, I remember.
Back when I was young.
As was I. Okay, so as Brian said, really, today I'm going to talk about just one part of research. I'm doing quite a lot of work on using non-model organisms, so using various birds to look at aging and early life sort of stressors, doing some work in humans with collaborators, some stuff in methylation clocks as well. But today I'm just going to talk a bit about genetic pathways underlying or modulating aging. So I'm going to talk very briefly on aging and disease, which I think this audience should all know. Then I'm going to talk a little bit about previously published work looking at targeting the insulin IGF-1 signaling pathway in mammalian or mechanistic target of rapamycin pathway. Some recently published data looking at RNA polymerase 3. And then I'm going to finish up just talking a little bit about mouse genetic background and longevity, which I think is a really important point, which has been ignored a little bit, I think, in the past. So really, I'll start with very basic sort of background. So as we know, we're living longer. The elderly populations are making up a greater and greater proportion of society. This is just some data here from Singapore. So in 1960, if you're a man in Singapore, your life expectancy was about 60 years of age, and a female, you were about 62 years of age. And you can take this forward now, 2019, Men lived for 81 years and women nearly 86 years. Been a slight blip post-COVID, but we all know that people are living longer and longer, and this has obviously important implications for society, healthcare, social care. And this is because age is associated with functional decline and multimorbidity. This is just data here from Hoffman and colleagues showing age along this axis here, and this is just the instance of various pathological diseases. It's estimated that people over the age of 65, probably about 2/3 of them suffer from multimorbidity. So they're suffering from 2 or more of these age-related diseases. You can see here these diseases are primarily in old age related to vascular disease and cancer. And this is just to show there's conservation in these aging phenotypes, slightly different diseases in dogs dominated by cancer. But again, you're getting this age-associated increase in multimorbidity. So when we're trying to study aging, we all know that aging, or the aging process itself, is really complex. We've got influences of genetics, we've influences of diet, where you live in the world, how rich or poor you are, and we've also got the effects of differences between males and females. Females typically live longer than males, and you've also got the effects of chance of stochasticity. And these all act to affect our aging rate. Some individuals live longer than others, and we also know this occurs in the model organisms. So here I'm speaking of things like C. elegans, Drosophila, and mice. And we've known in the case of caloric restriction for over 100 years now, and we know now with genetics, exercise has effects. We also know that certain drugs acting on the pathways I mentioned, such as insulin, IGF-1 signaling, particularly the mTOR signaling pathway, we can intervene to modulate the aging process, particularly in model organisms. So aging is complex, but we're starting to be able to intervene to modulate this. And this complexity is just shown again by this graph here. And this shows life expectancy in 2 parts of Glasgow. And this is from 2005. So these individuals, these are for men in Glasgow. So Glasgow Lenzie is an affluent part of Glasgow. It's to the north of Glasgow. So if you're a man in Lenzie, your life expectancy in 2005 was well over 80 years of age. So it's actually in excess of long-lived countries such as Japan, Singapore, and Iceland. And it was far in excess of the British average for a man. But if you're a man who lived in the east end of Glasgow, Glasgow Carlton, your life expectancy was around about 55 years of age. And this always amazes me when I show this, because this shows the distance. Glasgow's a city of about 600,000 people now, and the distance between Lenzie in the north and Glasgow Carlton, which is the east end of Glasgow, is just under 9 miles. So in this distance of just under 9 miles, your life expectancy is changing by nearly 30 years. And this is thought to be driven by lots of factors, but socioeconomic deprivation appears to be really important. So we know that this idea of the geoscience approach to aging, that if we can modulate the aging process, slow aging, we hopefully can preserve late life health. So what we're trying to do is to compress that period of life where we're suffering from multimorbidity. So it's really targeting the aging process to then delay or in some cases prevent these age-related pathologies. And from earlier work, which were roundabouts of the early 2000s, this was work I undertook in London. And this was a large-scale study driven primarily by Linda Partridge with David Gems and Dominic Withers. And Linda was driving the work in Drosophila, David in C. elegans, and Dominic in mice, and using this 3-pronged approach to try and understand the genetics underlying aging. And we were particularly interested in these pathways are involved in sensing nutrient status. And what these pathways do is they match metabolically costly activities. These are things such as development and growth, fecundity, stress resistance. So they're matching these metabolically costly activities to nutrient levels. Okay, so if nutrient levels are high, these pathways are activated. And we were particularly interested in components of this pathway. We're interested in insulin receptor substrate proteins. So these are essentially sort of cytoplasmic adapter proteins, which are involved in propagating. So you get activation of the receptors, IGF-1 receptor, the insulin receptor by their appropriate ligands. Then you get the adapter proteins act to propagate. They become tyrosine phosphorylated and you get propagation of the insulin signaling cascade. So we're interested in these, and this was driven by work in Drosophila, which has a single IRS protein known as Chico. This was work by Lyndon David, who showed that if you knock down Chico in flies, you extended lifespan. So these are the Chico flies here. Other thing to note, these animals typically, with lots of these pathways, because they play a key role in growth, these long-lived animals are typically dwarf. So we're interested in IRS1 and IRS2, and we're also interested in ribosomal S6 kinase, which is the main downstream effector of mTORC1. Okay, so we did long-term lifespan studies in these animals, and we showed in 2 independent studies, if you knock down or knock out IRS1 protein in mice, so this is from embryonic development, you're knocking out IRS1 in every single cell within the body of these mice. These mice, as I say, are dwarfs, so this is an IRS1 knockout. This is a sister here, so you can see they're really small mice, but you get lifespan extension. So these are the IRS1 mutants, and the blue, in all cases, a show of the wild individuals. So you've got an increase both in median and in maximum lifespan. And we did a subsequent study in males, an independent study, and we showed in the males we also saw lifespan extension. This is IRS1. IRS2 is quite distinct. If you knock out IRS2, excuse me, the mice are short-lived. And this is because IRS2, rather than IRS1 has a major role in post-embryonic growth, IRS2 has a major role in beta cell function, and particularly beta cell expansion under conditions of glucose sort of dysfunction or intolerance. And IRS2 mice are unable to increase their beta cell mass, and they ultimately become overtly diabetic. And you've got sexual dimorphism here. The male mice are really short-lived, lived for about 100 days on average. Females live to about 500 days. We also were interested, as say, in S6 kinase, and we again showed if you knock out S6 kinase, this is combined sexes, again, the blue are the wild type, the knockouts are red, you get lifespan extension. This is combined sexes, this is for females. For the males, we didn't get lifespan extension, and I think the case here, we're slightly underpowered in our mouse study here. There was a subsequent study by Paul Fox and colleagues where they're looking at a downstream effector of S6 kinase known as EPRS, and they essentially replicated the ribosomal S6 kinase knockout studies, and they showed that there was life extension both in male and female mice. And there's lots of other studies now. We know that lots and lots of people are looking at rapamycin, which acutely inhibits mTORC1. And there's lots of other studies now showing that this pathway is really druggable and has major effects on longevity in model organisms. But really, as I said, what we're interested in is that we're trying to preserve or maintain late life health rather than simply extending lifespan. And this graph here again shows this is a IRS1 knockout mice, and this is a sister. So these mice are about 700 days of age. You can see this little knockout is bright-eyed and bushy-tailed. The sister here, the wild type, you can see alopecia here. You can see probably evidence of cataract. You have the classical kind of splayed gait, old mice, and you've got curvature of the spine. So these mice are essentially, this is typically for a wild type mouse, the C57 black 60 background. This is the knockout. And we've shown in our studies that these knockout mice are longer lived, but they appear to have better cognitive function in old age. There's evidence of hippocampal neurogenesis. They've got better glucose tolerance as they age. They've got better cardiac function. They've got some evidence that they've got preserved immune function. They remain leaner. Mice, like humans, get fat as they get old. These mice are protected against this age-associated adiposity, and they're also protected against sort of classic age-related diseases associated with the strain of mice. These are things like cataracts, idiopathic dermatitis, which I'll mention later, and osteoporosis. The challenge, though, is really trying to understand what is driving these changes in aging rate in these animals. And this is a really difficult thing to do. And I think the mouse aging studies has slightly suffered from first past the post mentality, that there's a real drive to sort of publish long-lived mouse mutants, but really trying to understand what is actually happening mechanistically is a bit more difficult. And a large amount of my research over the past few years has been trying to look at this using sort of transcriptional approaches, metabolomics, various other things to try and understand from a mechanistic perspective. And this is just some work here that we've done in the SS kinase relatively recently, where we've looked at hematopoietic stem cell function, and we can do these sort of recipient competitive assays where we irradiate recipients and we put in these CD 45.2 and 45.1 cells, and we'll look at the competitive ability. And what we've shown in SS kinase mice, we've unpublished work from IRS1, is that the hematopoietic stem cells, not when these animals are young, but when they're older, have got enhanced sort of competitive ability. And they also, the stem cells appear to remain quiescent for longer in these animals. So there's lots of ongoing work here. I'm now just going to show some recently published work where we've been looking at RNA polymerase 3. So as you know, there's 3 RNA polymerases in mammals. You've got POL1, POL2, and POL3. And POL3 is involved in the transcription of non-coding RNAs, things like 5S ribosomal RNA and tRNAs and things like that. So we were interested in this from an aging perspective again. And once again, this was driven by work that was done in invertebrate models. And this was work from the Z. Falitz group in London and Jenny Tillotson in Kent. And they published a paper in Nature 2018, where they showed that if you knock down either through RNAi or genetically POL3 in these invertebrate systems, you extend lifespan. So these are the vector, empty vector, and these are your RNAi. And you can see here, both for the fly and the worm, you've got a longevity phenotype. And they also showed that if you knock out POL3 only in the gut, or in the case of the flies, only in intestinal stem cells, you've got recapitulation of this longevity phenotype. So we were interested in looking at this in the mice to see again if there was conservation. So this was work again with Jenny Tillott, who works in C. elegans, Nazif, who works in Drosophila. At the time, it was Gillian Borland and Stephen Wilke who worked in mice. And we targeted at the time POLR3b, which is the second largest subunit. Forms the catalytic core of POL3. And unlike the other studies, this was a CRISPR-Cas9 mediated mutation. So it's an 8 base pair insertion here, which caused a frame shift mutation and early truncation in the POL3. And we did this, these mice were heterozygous for this. So one allele had this sort of insertion. If we did two, so we made the mice homozygous, it was embryonically. So we looked at this in the context of longevity, and very depressingly, we showed that in our hands, POL3 didn't limit lifespan. So these are combined sexes, again, the wild type, and in blue again, the knockout, the heterozygous knockout here are in red. So these combined sexes, females and males. This is not significant here for males. I also should note, which we're going to talk about, is that these mice were in a C57 black 6N background, as opposed to C57 black 6J, which our other previous imaging work had done. We saw no evidence of differences in tumours in these mice. We then looked at a number of parameters related to health, where they, although they weren't long-lived, were there some preservation and health in these animals. We looked at gut health, and this is from the work showing that in the flies, you have the guts are leakier, and this is wild-type individuals compared to the POL3 animals. So the POL3 animals had preservation and gut function in female flies, and this is a study where you give the animals a diet with blue dye in it, and you essentially get, if you've got a leaky gut, the blue dye goes into the organism itself. We saw, if we looked at females, the wild type in blue here, the red are the heterozygous knockouts. The female mice actually had leakier guts in contrast to the flies. The males had a preservation in gut health in old age. And we saw, although we didn't see much in the way of genotype differences, we saw a huge number of differences in terms of comparisons between the sexes. We looked at bone health. As I said, mice suffer from osteoporosis as they age. And we actually saw in middle age that the POL3 females actually had a preservation. So this is just a cross-sectional micro CT through the bone of the femur. This is the trabecular, the honeycomb system here. You can see it's preserved in the POL3 compared to the wild type. We looked at a host of other parameters. We didn't really see any difference. This is just grip strength here at various ages. We see no difference in old age. In 18 months, actually, the females had better grip strength compared to the males when you can normalise to body mass. But we didn't see anything with advancing age, and we saw no difference between genotypes. The one thing we did see is that the POL3 heterozygous females were much more prone to idiopathic or ulcerative dermatitis. So this is an image here, a really horrible condition here. And we saw that these female mice had a higher incidence, and they also, the age of first instance, they got ulcerative dermatitis slightly earlier as well. And we've looked at this in more detail, and we've seen that these female mice have higher plasma IL-6, they've elevated expression levels by IL-6 in their skin, and also in culture dermal fibroblasts. They've got increased collagen content, but kind of confusingly, the wound healing appears to be more rapid in these mice, so we're kind of still ongoing with this to try and understand it a bit better. We also looked at frailty, so we looked at a whole host of different parameters. This is based on the Howlett and Rockwood mouse clinical frailty. And again, you can see that frailty increases with advancing age. Again, we saw no specific genotypic differences in between our different males and females. He's slightly more frail in the males here, but it's not significant. One of the issues with frailty is, as the animals get older, you obviously have animals, your sample size decreases, so it becomes much noisier and much more difficult to pick out specific differences. Okay, so for the last couple of minutes, I'm just going to change tack very quickly and just talk a little bit about genetic background in aging. And really, as people working in aging in mice, we've really been over-reliant on inbred C57 black 6J animals. Particularly, we've been focused on male mice. And this then leads to this idea of private-public mechanisms of aging, which was first proposed by Jens and Partridge a couple of decades ago. So private mechanisms of aging are private to that organism or that strain. Public are things conserved. And So we've lots of the majority of the work in aging has used C57 Black 6J. And this denotes Jackson Lab. They moved to Jackson Lab in 1948. C57 Black 6N, 1951, National Institute of Health. And all these populations of mice, wherever they are susceptible to a whole host of different things: genetic drift, founder effects, inbreeding depression, laboratory adaptation, and mutation accumulation. And there's lots of phenotypes associated with these two substrains, 6J relative to 6N, that change. And lots of these are sort of phenotypes we measure with aging. So 6J have impaired glucose homeostasis relative to 6N. They've enhanced motor coordination, improved vision, higher blood pressure, things like that. So we need to be careful when we're thinking about mouse models and longevity. And this is just some work here. So these are from independent studies from the work in 6J previously. So this is just looking at two different animal conditions, or one in London, one in the UK. But you can see here the significant differences, particularly in females. So these, the red line are the 6J and the blue line are the 6N. And there's also some work from the Jackson Lab looking at this as well, similar. So 6N mice tend to be much shorter lived than 6J mice. And this is important because the International Knockout Mouse Consortium, where we got these CRISPR mutant mice from, they're going to be generating knockout mice, and they're trying to do this for every gene in the genome, and these are all going to be in C57 Black 6N background. There's been a lot of criticism previously, this paper here by Holzenberger, where they looked, this was really the first example in the insulin signaling pathway of knocking down a gene, IGF-1 receptor. You got lifespan extension, but there was criticism at the time that this strain was relatively short-lived in the wild type. And this strain is relatively short-lived. They looked at this again in C57 Black 6J. They still got significant lifespan effect, but the effects are much, much more moderate. And we've also we looked obviously in C57 Black 6J. And the Partridge group has now looked at this again, IRS1 mice on a hybrid background. And again, they see lifespan extension. But these are really long-term studies and difficult to do, I think, for lots of people. And I think this, I'll just finish this slide. This is really fantastic work from Beverly Pagan and colleagues in the States. And they've looked both in ad libitum diet and caloric-restricted diet at genetic differences in lifespan. And there's actually huge variation in wild-type strains of mice. It's actually 3 times greater between these different strains. It's much bigger than the longest lived mutants reported. I think there's a lot of opportunities there to maybe try and look at this in a bit more detail. Okay, so I think that's my time up. So thanks for listening, and I'm happy to take any questions.
Thanks, Colin. A lot to talk about there. I want to come to the specific points of your talk, but maybe I'll start on a broader point. Since you're a mouseologist, and there's been a lot of debate recently about whether mice are a good model for humans or not. And, I've kind of made the public argument that some of these artificial disease models may not be good models for human disease, but that aging in mice is looking at the natural process may be more relevant to the natural process in humans. What do you think about this whole question and that conjecture?
I'm probably biased because I work, as you say, in mice. I think
so am I.
I once got a grant review, and there was one line that said mice, not men. And that was, but they're not perfect. I think, there's all this discussion that maybe we should look at wild-derived mice, and, we need to, these inbred strains, maybe, the stuff that Rich Miller's doing with these 4-way crosses, maybe at least, because humans are not, genetically uniform. So I think they're probably, at the moment, the best model we have. They're genetically amenable. But I think, people tend not to use rats, for instance, which, I think is just a cost thing, has been historically. So I think there is good a model as we've got at the moment, but as you say, some of the disease models may not be perfect. But I think what we do know from the context of aging is the phenotypes associated in aging are really highly conserved from C. elegans through to mice, through to humans. So I think they can give us insights for sure. But I think the challenge is going from mouse research to translating it, because everyone wants this, fundamental research to be translated. And I think there's still obstacles to that I think is proving hard to get past.
Have you tried looking at the methylation clocks in mice?
We've not. We've just published recently published some methylation clocks in humans with Paul Shields and a group in the Karolinska looking at it was kidney disease and looking at methylation clocks. So we are actually doing some stuff at the moment, Paul Shields, by looking at mice with particular interventions, drug interventions to look at methylation. But that work's ongoing, so we haven't actually analysed it.
Yeah, the Glasgow data is quite striking, and I don't think it's particularly unique to Glasgow or Scotland. If you look at the wealthiest counties in the U.S. versus the poorest counties, there's huge gaps in life expectancy. And probably what I think this is a measure of the quality of social support, education, healthcare that exists in various countries, and what it really is an indictment of the difference in accessibility to different classes in Western society. I think it's scary numbers to look at, though.
Yeah, I think it is driven by socioeconomic status, primarily access to, good diet, and there's been a lot of work in Glasgow looking at these, comparing these different areas. And, if you're in a deprived background, your levels of systemic inflammation are higher. They've looked at telomere shortening. They've looked, lots of things are driving this. And I think the scary thing is it's not getting any better. But I don't think it's certainly not limited to Glasgow. I think you look at many of the places across Europe and North America, I think it's the same.
Yeah. I think, you kind of brought this up. I'm making a jump back to mice here, but, you can look at all these different measures of aging and they become entrained to each other. And so you mentioned telomeres will be shorter and methylation clock will be higher and inflammation will be higher, et cetera. The same thing is, I think that gets to the question you brought up of how to get to mechanism in mice or really anywhere. When you intervene with something like mTOR pathway, all of the pillars and hallmarks of aging are affected. And we've struggled a lot trying to understand the proximal effects of any particular intervention in longevity. How do you go about solving that problem?
I think people are always asking you, okay, so what's a good assay for aging? What is this intervention doing? You look back at caloric restriction, it's hitting so many different targets. And we've known, the work by Osborne in 1917 showing that if you reduce food intake, you can extend lifespan, and the work by McKay and colleagues. And we still don't really know how caloric restriction or dietary restriction works. To be honest, I'm not sure, Brian, how, because it's getting away from what's correlational and what's causative. And I think these interventions are going to tweak lots and lots of pathways and mechanisms. I honestly don't know how you get to the crux of that.
Yeah, I think you're hitting like nodes in a network. And, I would point out, I'm sure you're aware of Sheen Tsai's work, here, used to be my postdoc in Seattle and now professor here at NUS. And she's also tried to go downstream of mTOR and look at a 6 kinase knockout and 4-ABP activation. And, you find you can bifurcate some of the benefits of mTOR either through 4-ABP or S6K, but you start to dissociate the effects, and then then you don't get the broad effects of mTOR. So I feel like the mTOR itself is kind of a node in this network, and if you hit that node, you get all of the benefits. If you go upstream or downstream, you get some, but not all of them. So.
Yeah, I think that's the challenge, and I think whether it's not whether you need to understand the mechanisms, but if you have something that is acting to slow aging, that hopefully if you can translate it, that is important. But I want to understand the mechanisms. That's what I'm interested in. I think it's challenging.
Yeah. For sure. The Pol3 data is quite interesting. And Pol3 is, if I recall, is doing transcription of tRNAs and small ribosomal RNAs. So it's really involved in translation and ribosome biogenesis. A lot of longevity pathways, including mTOR, have been linked to that. And also, I think we tend to forget that mTOR regulates transcription as well as translation. And this feeds right into Pol3, I think. What's the take-home message from the Pol3 seems to be similar. There's some benefits and some things that are not good maybe with aging. But is that, it's really kind of a dosage question too, right? Because you can't knock out the gene completely in a proliferating animal like a mouse. I think, and so is it maybe just didn't get the right dose?
I think possibly. We saw, you got a reduction in Pol3 protein levels and transcription. I think in hindsight, that was the mouse that was available. This study was started 5 years ago. I think possibly we should have targeted Pol R3A rather than 3B, and maybe we'd have seen, because some of the work coming out of the flies, which is much more amenable to genetic modulation, are starting to show that may be a slightly more important subunit. But I think it is. I think it's possibly we did decrease protein levels expression, but I think there was possibly compensation, because I think it's so fundamental that we didn't see any effect in lifespan, and the effects on health span were, kind of all over the place. It was a bit depressing after you spend 5 years to do that.
Always the fear. You go to all the trouble to set up a mouse longevity experiment, and you don't get.
So it's not the first time it's happened. So you can't do anything about it. It doesn't lie.
Well, one thing it does though is it proves you're honest in the field. So
yes,
we need more of that, I think.
Yes, I totally agree.
It also reminded me a little bit of the work on the nucleolus that we did a little bit, but Adam Antebi's really done a lot of that, and others. Yeah. And there is a connection, I think, between ribosomal processing and synthesis and longevity. It's just really hard to pin it down and hit it at the right way, I think.
Yes, I absolutely agree.
I was also curious about in the S6K1 knockout, the hematopoietic stem cell robustness. I think in humans, that I'm more and more believing that the hematopoietic stem cell pathway is really probably one of the big drivers of longevity. So it's interesting to see the S6K have that phenotype. Have you looked at other properties with aging in terms of lymphoid and myeloid transitions and maybe even chip?
So in that paper we looked at, we've done a little bit of myeloid. We're thinking about, with a colleague here who works with stem cells, not in S6 kinase, but another mutant associated with that, starting to look at that. I think it's potentially really important. We've got unpublished data in IRS1 showing a similar sort of phenotype again. So I think these sort of robustness of your stem cells, and I think is particularly, it makes sense. It's just difficult to do. And I had a meeting today with them, and I think the issue is that stem cell biology, to do it properly, is really difficult. So you have to have a stem cell expert to kind of look at this. They work on rapamycin and stem cells, and the sort of vaccination response. Yeah, it's really interesting, and I think I think it is. I think it's an important and interesting area, but I think to do it properly is difficult.
Yeah, and I think there was old data, at least and maybe newer data too, that rapamycin preserved hematopoietic stem cell function with aging, right? That was one of the first papers that had longevity data, if I recall. So,
That's right. That was and they challenged the mice with influenza, and I think, or
yeah.
I can't, yeah. So, and there was work before that. There's a lot of work in the US looking at, I can't remember who did it now, but they're looking at caloric restriction and hematopoietic stem cell function things, showing similar things again. So there's data out there. I think but as I said, I think the challenge is with a lot of lifespan work, it's, you have a long-lived mutant, you get in a high impact journal or a long-lived intervention. If you don't, it's sometimes a bit difficult to publish it. And in a way, people are less interested in the fundamental mechanisms. They want the high impact paper, and I think that we suffer a bit in the aging field from this first past the post mentality.
Well, I could talk forever. I think instead I'll bring Anna in, partly because there's a lot of questions from the audience, and partly because I think she has the coolest hair of anybody on the show. So,
Brian. And thanks, Professor Selman. That was such an amazing overview of these longevity genes, and your amazing work on mice. I think the audience is also quite inspired in terms of questions. And as there were especially many questions about the differences in sex that you also presented in mice, but for example, one member of the audience points out that supercentenarians are almost exclusively ladies. So they were wondering whether there were any anti-aging or longevity genes located on the X chromosomes.
So there is, I think IRS2 is, but it's not a longevity gene. I wouldn't know offhand about that. I mean, there's been lots of work in centenarian studies looking at genes within this pathway that are associated with longevity, and you get hits for lots of them, mTOR signaling genes, insulin signaling genes. We've actually just done something looking at POL1 and POL3, and we also get hits in that, but we don't seem to replicate that in the mice. I think there will be. I mean, I think with the mice, one of the potential explanations is that you're getting, these mice are typically subfertile. And I think, of course, when you're doing lifespan studies, you're not breeding the mice, but I think you possibly the effects or the impacts on fertility are much greater in females than males because the cost of reproduction is much, much higher. But nobody, to my knowledge, has looked at this in the context of aging, really, in these long-lived mutants. But really interesting question.
Right now, what are the best candidate genes for sex-specific effects or dimorphism?
Again, certainly lots of the insulin signaling, the mTOR as well. Rapamycin has sexual dimorphic effects. A lot of this, again, I think is driven by differences in genetic background as well. And mice are kind of a bit peculiar, particularly C57 black 6J, where the males are longer lived than the females typically. So, I think you've got lots of targets in insulin mTOR pathway, but I wouldn't know one particular gene at the moment. I have my biases, but I think most of these pathways seem to have sex specific. That's why it's important to look at both males and females. And now things have changed, certainly in the US now and in the UK as well. You can't do aging studies unless you're looking at males and females. I think it's really important.
It was just jumping in. Sorry. It was shocking that when we first started looking at diabetes studies because of 4EBP, we saw benefits in females and nobody, I said, where else do you see that? And almost nobody in the field was looking at females. It's amazing how much disparity there was in these animal studies.
Yeah, it's unbelievable. And And I think there's a study by Holly Van Remen years ago looking at, it's a kind of big meta-analysis looking at oxidative stress measures and things. And I think vast majority of studies were done in male mice and C57 black 6. So I think we're improving in that area that we're starting to study males and females. But I think, I mean, certainly with the POL3, we had far more effects or differences between males and females than any genotype.
Thank you. Just to switch topics a little bit, somebody is asking, is there something that mice are better at than humans? Wound repair, or maybe not getting particularly fat despite little exercise and available food 24/7? And they live quite a bit for their size. So is there something we can learn from mice?
I mean, I think we can learn, as I was saying to Brian, fundamental mechanisms. In terms of what mice are good at, the humans are not, I suppose, as being a mouse. I don't know, they're very, very good at reproducing. I mean, in terms of wound healing, I mean, when they're young anyway, but I mean, most mice, like humans, get fat as they get older. So it's just, I think one of the challenges, again, has been, we're talking about males and females. I think lots of the aging studies, and I include, I'm including myself here. I think lots of these studies has looked at, when they talk about aging studies, they look at mice at maybe 6 months of age, and they, categorize these as being old, and they're not. They're just kind of mature adults. And I think, we need to extend out what we're looking at in mice, to older ages and things. But this is difficult to do because it's expensive to keep mice. But I don't know what I think a mouse is good at being a mouse. It's quite a difficult thing to answer. But I mean, certainly lots of the diseases that are associated with aging are recapitulated in the mice and things like that. So,
they don't really get athero, so maybe they're good at keeping their vessels function.
Yeah, so I think that is one thing that they're particularly good.
Yeah, this is just my own follow-up question, but is it possible actually to look at these behavioral effects on gene regulation in mice? Because we understand that you can knock out the gene and then you can study those effects, but are there ways to study how the environment and lifestyle influences gene expression?
There are. I think the easiest thing to do is to put them on a high-fat diet and, so giving them excess calories. Do you mean in terms of sort of deprivation? Or, I think it's difficult because, certainly in the UK, our mouse experiments are really highly regulated. And I think one of the issues I didn't touch upon when we're looking at the frailty in mice, we have humane endpoints. Our mice aren't allowed to, we have to intervene if they get ill. And most of these humane endpoints are essentially the same endpoints as frailty. And I have looked at the frailty in our mice. So these are male C57 black 6N, and compared them to some of David Sinclair's work on mouse frailty. And the mouse frailty in his studies for a given age is much, much higher than ours. I think it is possibly to do with how we have to intervene from a legal perspective. It's difficult. I think it'd be really interesting, but I think it's difficult to do because you then, if you're kind of depriving these animals in some way or stressing them, it may be difficult to get guidelines, certainly in the UK, to get guidelines. I know there's people looking at, people have looked at singly housed compared to group housed animals under different contexts. There's some stuff, I think, going on in rats as well, looking at sort of increase the number in the cage, it's a difficult one though, because if you increase the number in the cage, there are benefits because they're social and they have better thermoregulation if they can huddle and things. So it's quite difficult to disentangle what is related to stress or deprivation. I think it's a really interesting thing, but I think it's quite difficult to do certainly under UK Home Office guidelines.
Yeah, as you mentioned, it's also clear that there are also these ethical considerations of what you can put the animal.
Yes, absolutely.
Yeah. Well, one member of the audience was wondering whether there is also some evidence that would be aligned with the evidence you presented, but in aging primates. So were there any primate studies?
I mean, there's some fantastic studies in the US where they've looked at caloric restriction. So they've got the Wisconsin group and the NIA have looked at rhesus macaques, and there's also some work in a little kind of lemur as well. But these are mainly in caloric restriction. But I think they're starting to look at methylation clocks and things like this in these primate studies. And again, they may be, under some contexts, a better model, but it's very, very difficult to do that. And you certainly can't do that work in the UK. So, it's difficult, again, very, very long term. I mean, these animals are living for 30, 40, 50 years. So it's difficult to get continual funding to look at this.
Yeah, for sure. And probably a question of cost and number of animals, because you can have a lot of...
Yeah. So, I mean, it's really powerful system, but I think there's only a few places that can do this type of work at that level.
Yeah, great. So one person is also wondering whether, how much you understood of the translation of these longevity genes into really the protein pathways and what they actually do within the mice.
I think that's really, really important. We know in these mice, we've done lots of RNA-seq studies showing that from a transcriptional level, there's major overlap. So if you compare the transcriptional signature in particular tissues in IRS1 with SS kinase or with dietary restriction, you get real clear overlap within a tissue, but you don't get any overlap within the same mouse between different tissues, which is because they're responding slightly differently. In terms of proteomic approaches, we've not done so much, but I think that's really important because what's actually happening, and I think there's, even with metabolomic type approaches, we've done some stuff between, it was IRS1 dietary restricted mice and Ames dwarf mice, and there was certainly less overlap between metabolite levels compared to transcriptional levels. So I think there's still lots we need to sort of find out. I think it takes time and money.
Yeah, we've got just a little bit of time. Is there one last burning question that we can get a short answer to?
Okay, so then maybe a bit more of a question about future, once this research would get translated to humans. The question is, how might the risk of cancer and the benefits of longevity extension techniques be safely balanced, keeping in mind that HSCs may trigger tumor development?
Well, I think that's the sort of $1 million or the $1 billion question, is how to translate. The big thing at the moment is repurposing drugs. We know that rapamycin, we know that trametinib, various other drugs all act in mice to extend lifespan, but generally, not proposing to take these drugs, certainly at the moment, until we know more. So I think there's lots of work needs to be done yet. And I think, I was just speaking to Brian, the jump from mouse to human is still a huge distance. I think a lot of work needs to be done.
Great. Well, thanks a lot, Colin, for joining the show. It's really wonderful conversation.
Thanks so much for the invite.
Anytime. I want really short announcements tonight. I want to remind participants to use the chat function and panelists and all attendees option to leave comments and feedback on the show so that everyone can view it. And I'm going to leave you with a final video about a 96-year-old who stays fit with yoga and dance. Thanks for joining us.
Please welcome 96-year-old Phyllis Sues to the show. Woo! Welcome! Phyllis does things with her body that I can't even think about.
Oh, yes, you can, dude.
Oh, no, I
Phyllis,
can't.
I'm just going to go out there and say that you are my hero.
Mine too. So,
unbelievable.
What is your secret to staying so fit?
Work. A lot of work.
And I think when I first started to dance, I really wanted to be skinny so that I would be I would be like a bird and fly away, and I could be lifted by the guys, because I did in so many Broadway shows, dancing. And I danced until I was 50.
Okay.
And then I opened a fashion designer, and
Nice.
for 22 years. And now I'm back dancing, so it's full circle.
Wow.
Can you give a few tips to people out there who maybe haven't been as active as they should have been? Movements that people can engage in that are not as advanced as you doing the splits?
I love what I do, and that's the key. You have got to love what you do.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.